Welcome to Tomorrow Unveiled. Today we're tackling a question as old as humanity. Can we truly beat aging? We call this deep dive the Immortality Update.
Yeah, that's the big one, isn't it? And,
Hmm.
for decades, talk about longevity, it was mostly, well, speculative, fringe almost.
Right, just theories.
Exactly.
Yeah.
But the consensus now, looking at the research compiled for us, it really confirms that 2025 it's a turning point, a breakthrough year. The whole conversation, it shifted.
How so?
It's moved from, abstract ideas and theories to actual validated clinical applications, things we can measure and see working.
Okay, so that's our mission for this deep dive with Tomorrow Unveiled. We're going to review the latest clinical trial findings in longevity science. Our dedicated research team pulled together all this material, and we're really zeroing in on the advancements reported this year. The aim is to quickly unpack, the most impactful molecular discoveries, the emerging treatments. And the goal isn't just adding years to life, right? It's about extending health span.
That's the critical distinction. Health span. The time we spend living vibrant, healthy, disease-free lives, not just existing longer.
Absolutely.
And that's what the data our research team gathered highlights. It shows this huge paradigm shift happening in the field. We're moving way past those vague sort of anti-aging promises you used to see everywhere.
Okay.
We're at this inflection point now.
Yeah.
It's where genuine evidence-based biology has actually met specific molecular targets.
Yeah.
And it's giving us measurable outcomes, things that, honestly, even 5 years ago, seemed like science fiction.
I like that emphasis on evidence. Rigorous measurement, not just, wishful thinking.
Exactly.
Okay, let's jump right in then. Let's talk about the core molecular strategies that seem to be fueling this whole movement. All right, so when we look at the source material our team compiled, there are these two compounds that just immediately jump out. They seem to be the real focus right now for cellular energy research. Nicotinamide mononucleotide, NMN, right?
NMN, yeah.
And resveratrol. The data keeps mentioning their synergy. This combination is a primary strategy for longevity. Why? Why these two together? What's the excitement about?
Well, the excitement really stems directly from understanding a core problem of aging, maybe the core problem. Cellular energy failure.
Okay.
Think about it. Pretty much every function in your body, whether it's repairing your DNA, your immune system, fighting something off, anything, it relies on this critical coenzyme. It's called NAD plus NRD, nicotinamide, adenine dinucleotide.
NAD plus NRAD.
Got it, right. And the issue is NAD plus levels, they naturally and actually quite steeply decline as we get older.
Okay, so they just drop off.
They really do.
Yeah.
And when your NAD plus levels drop, all those crucial cellular processes, they start to slow down. Your body's ability to repair damage just isn't what it used to be. It diminishes.
Okay, so if NAD plus is this fundamental resource, like the fuel for the cell, then we need a way to top it up, right, to replenish it efficiently.
Precisely. That's the goal. Now, NMN itself, it isn't NAD plus NANRD. It's what we call a precursor.
A building block.
Exactly. Think of NMN as the highly bioavailable raw material. Your body takes it and uses it to synthesize more NAD plus ferrol. So you're essentially topping up those critical cellular energy reserves.
Makes sense. And where does resveratrol fit into this picture?
Resveratrol's role is a bit different, but it's incredibly complementary. It acts as an activator for something called sirtuin pathways.
Sirtuins, I've heard of those. They're like longevity genes.
Sort of. Think of them as the guardian proteins for your genome. They're crucial for things like DNA repair, managing inflammation, regulating cellular stress. Really important stuff. But here's the catch. Sirtuins are completely dependent on NAD plusly. They need high levels of NAD plus to do their job properly.
Ah, okay, so the synergy becomes really clear then. NMN brings the fuel, the NAD plus row, and resveratrol acts like the accelerator pedal. It turns on this crucial sirtuin repair crew, making the NAD plus fuel work harder, more effectively.
That's a great way to put it. NMN provides the resource. Resveratrol optimizes its use by activating these key protective pathways.
But do we have solid proof? I mean, specific convincing data showing that this combination is actually better than just taking NMN by itself.
Oh, absolutely. The clinical consensus really confirms this synergistic power. It's real, and it's highly measurable. The research team reviewed data, including some key animal studies, which demonstrated this effect very clearly.
What did they find?
When NMN and resveratrol were combined, the boost to NAD plus levels wasn't just minor, it was significant. They saw a 1.59 times increase in heart tissue NAD plus mark.
Okay, 1.59 times in the heart.
And get this, an even higher 1.72 times increase in skeletal muscle NAD plus marlone. And this happened within hours.
Wow, 1.7 2 times in muscle tissue within
Yeah.
just 6 hours. That's pretty remarkable.
It is.
Functionally, what does that actually mean for the subject? I mean, it's not just a number on a lab report, right? What does that rapid boost do?
Yeah, good question. It translates directly into tangible benefits at the cellular level.
Okay.
We're talking enhanced mitochondrial function, better energy production, and also accelerated DNA repair velocity.
So the cells work better and fix themselves faster.
Exactly. Specifically in muscle tissue. This means the cells can manage energy production much more effectively. They can clear out metabolic waste products more rapidly. The implication there is improved physical resilience, better endurance.
Which lines up with what people report feeling.
Precisely. It's
Right.
exactly what the subsequent human trials started to validate. You see, a cell with adequate NAD+ and activated sirtuins, it just functions more like a younger cell. It repairs itself more efficiently.
Okay, let's talk about those human trials. The data our team looked at confirmed NMN safety across a really wide range of daily doses. You mentioned from 100 milligrams up to 1,250 milligrams. That's a huge difference.
It is a very broad range, yeah.
Does the research explain why there's such a wide safety margin and maybe who benefits from that really high end, the 1,250 milligram versus someone on the lower end?
That broad safety profile is actually one of the most reassuring findings. It's been corroborated across multiple studies reviewed by the research team, and it gives significant confidence for, future development and potentially regulation.
Mm-hmm.
As for why the range is so wide, the data strongly suggests it comes down to individual metabolic differences. Things like your body weight, your current age, certainly your existing level of metabolic stress. These all seem to dictate what the optimal dose might be for a specific person.
So it's personalized.
Very much so. Generally speaking, those higher doses, the 1,000 milligram plus range, they're being explored more in subjects who might be dealing with specific metabolic challenges. Or perhaps those aiming for a really maximal boost in NAD+ to see measurable functional improvements, like in athletic performance studies.
Okay.
But importantly, even the lower doses, which you might call maintenance doses, they proved to be safe and still offered notable improvements in the trials.
What kind of notable improvements are we talking about? What did the human subjects actually report experiencing?
Yeah, beyond just the raw molecular markers, like seeing NAD+ go up, the human trials started revealing palpable benefits, things people could actually feel.
Like what?
Subjects frequently reported a marked improvement in physical performance metrics, things like endurance testing, strength maintenance over time.
Okay, actual physical capability.
Yes, and also just a widespread increase in their overall sense of vitality, energy levels, general well-being. People just felt better.
That's huge. That moves the discussion past just, stabilizing some molecule inside a cell and into benefits that actually enhance someone's daily life.
Exactly. It validates the core strategy. Replenish this crucial coenzyme, get the repair pathways working, and performance, both cellular and systemic, improves. It's quite logical when you break it down.
Okay, so we've talked about the intervention, NMN and resveratrol, what we might take. Now let's shift to evaluation. How do we actually measure success? Because we know chronological age, how many birthdays you've had. It's pretty misleading in longevity science, isn't it?
Oh, absolutely. It's becoming almost irrelevant in these contexts. Your calendar age just doesn't tell the whole story. You could be 60 chronologically but have the cellular health of a 40-year-old or, well, vice versa.
Right, so the number of birthdays doesn't matter if your cells are effectively decades younger or older.
Precisely. And the research team specifically noted that refining the tools we use to measure biological age, your body's functional age, is a major ongoing focus in the field right now. It has to be.
Well, it has to be.
Well, think about it. If we claim a compound like NMN or an intervention like exercise is working, if we say it's anti-aging, we absolutely need objective, quantifiable proof. We need to show that it is successfully slowing, maybe even pausing, or potentially reversing the aging process at a cellular and systemic level.
We need to move beyond just people saying, I feel better.
Exactly. Subjective reporting is important, but it's not enough for rigorous science or clinical application. We need hard data.
So what we're really looking for is a genuine molecular clock, something that tells us the true age of our cells and systems.
That's the goal.
And the research material our team gathered, it highlights 3 specific sort of cutting-edge pillars of personalized testing that are being refined right now. Blood tests, epigenetics, and the gut microbiome.
Let's try and dissect these. Maybe start with the one that sounds the most precise. Epigenetic profiling?
Okay, yeah. Epigenetic profiling is it's widely considered the undisputed gold standard for measuring biological age today. It really focuses intensely on something called DNA methylation.
Methylation. Okay, what is that?
Right. So if you think of your DNA as the hardware, the fundamental instruction manual for your body,
Mm-hmm.
then methylation is like the software, or maybe like sticky notes attached to the manual. It involves these tiny chemical tags, methyl groups, that get attached to your DNA itself. And these tags dictate whether specific genes in that area are turned on or off. They control gene expression without changing the underlying DNA sequence.
Okay, and what does that have to do with aging? How does it tell time? Well, here's the fascinating part. As we age, these methylation patterns, these sticky notes, they change. They become sort of disordered. But crucially, they change in a highly predictable, measurable way across our lifespan.
Ah, so the pattern itself is the clock.
Exactly. The specific pattern of methylation across hundreds of thousands or even millions of sites on your DNA acts as a genuine molecular clock. It's literally keeping time inside every one of your cells.
Incredible.
So if an intervention, let's say it's NMN, or maybe caloric restriction, or exercise, if it's genuinely working to slow or reverse aging at a cellular level, the researchers expect to see those DNA methylation patterns shift. They should start looking more like the patterns you'd see in a biologically younger person.
So it's an objective sign that cellular function is actually improving, written right there in the epigenome.
Precisely. It's about as close to reading the cell's true operational age as we can currently get.
That level of granularity is just amazing. But okay, how do the other two pillars, you mentioned the blood tests and the gut microbiome analysis, how do they fit in? How do they complement this epigenetic data?
Right. They provide different but equally valuable perspectives. The current generation of blood-based age tests, they use a completely different approach. They're not looking at methylation. Instead, they look at a whole panel, often over 70 specific biomarkers simultaneously.
70? Like what?
Things like various hormones, key inflammatory markers like C-reactive protein, measures of liver function, kidney function, glucose control, lipid profiles, a whole battery of texts run at once.
Okay, so a really comprehensive snapshot.
Exactly. The algorithms then synthesize this huge amount of data to give you a dynamic picture of your current metabolic health, and importantly, your level of systemic inflammation right now. So while the epigenetic clock tells you the cell's sort of deep underlying programming status.
It's history, almost.
Yeah, kind of. The blood panel tells you how well your body, as a whole system, is coping with everything right now. Is it running efficiently, or is it under stress and inflamed?
Okay, that makes sense. Epigenetics is the deep clock. Blood is the current system status. And the third pillar was the gut microbiome analysis. How does that connect to aging?
Ah, the gut. It's a massive piece of the puzzle, and honestly, often overlooked until relatively recently in the context of systemic aging. We now know the gut microbiome, the trillions of bacteria living in your digestive tract, has a profound influence.
How so?
The analysis techniques are getting really sophisticated. They now look at over 30 different features, things like the diversity of microbial species present, the relative abundance of beneficial versus problematic bacteria, markers of gut lining integrity, digestive efficiency.
And what's the link to aging?
The research confirms a powerful, measurable link between a lack of diversity in your gut flora, an unhealthy microbiome, and increased chronic, low-grade inflammation throughout your entire body.
Ah, inflammation again.
Exactly. It's a major driver of most age-related chronic diseases. So by analyzing your gut microbiome, we can identify imbalances that might be contributing to systemic inflammation. And crucially, by addressing the gut through diet, probiotics, prebiotics, you can directly influence that inflammation and consequently positively impact your biological age markers across the board.
Wow. Okay, so it really paints a picture of aging as this complex interplay. You've got the drift in the genetic instructions, the software glitches, that's the epigenetics. You've got the metabolic wear and tear, the system stresses, the blood markers, and then you've got the environmental and lifestyle impact heavily influenced by what's happening in your gut.
It's a perfect summary. It's multifactorial.
So how does having all this incredibly granular data from these three different angles actually benefit the individual? How does it change things practically?
This granularity.
Right.
This is the absolute foundation of true personalized longevity medicine. It's moving us beyond guesswork because we now have these three distinct objective views of your biological status. Interventions are no longer just shots in the dark. For example, imagine someone's epigenetic markers show they're significantly older than their chronological age, but their gut microbiome looks relatively healthy.
Okay.
In that case, The treatment strategy might focus heavily on things known to impact epigenetics directly, like NAD plus enhancement with NMN, perhaps Senolytics, which we'll get to, specific types of exercise.
Right, targeting the epigenetic clock itself.
Exactly. But now imagine another person. Their epigenetic age is fine, close to their calendar age, but their blood markers show really high inflammation, and their gut analysis reveals poor diversity.
Different problem, different solution.
Precisely. For them, the strategy must include significant dietary interventions, targeted probiotics, maybe stress reduction techniques, things focused on quenching that systemic inflammation and healing the gut. NMN alone might not be the primary lever to pull for them initially.
So treatments are being precisely tailored based on an individual's unique molecular profile. We're moving away from just giving everyone the same advice or the same supplement.
Yes, it's about moving optimization away from population averages, which might not apply to you at all, and into validated individual responses based on your own biology.
This really does feel like moving away from, just throwing supplements at the wall and hoping something sticks.
Yeah, this supplement scattergun approach.
Right, and moving into a rigorously monitored, genuinely tailored approach to optimizing health and potentially slowing aging. But I have to ask the practical question here. Are these kinds of multimodal tests, especially the really detailed epigenetic profiling, are they even accessible or affordable for the average person right now? Or is this still mostly in the realm of high-end research and, longevity clinics for the wealthy?
That's a crucial point, and it's definitely a challenge that gets consistently raised in the industry reports our team reviewed. You're right, currently, the most comprehensive epigenetic profiling, the kind that looks at millions of sites, is still relatively costly. It's often still primarily used in research settings or specialized private clinics.
Okay, so not quite mainstream yet.
Not quite mainstream for the deepest level of analysis, no. However, the data also confirms that the technology is advancing incredibly rapidly. Costs are coming down significantly year on year. There's a huge push to develop more targeted, affordable epigenetic assays.
What's the goal then? Where's this heading?
The goal, which is pretty clear across the sources, is to eventually integrate simplified but still accurate epigenetic assessments, perhaps focused on the most informative methylation sites into standard primary care, maybe within the next 5 to 10 years.
Wow, okay. Part of your annual checkup, maybe.
That's the vision, making this kind of personalized, preventative insight universally accessible rather than it remaining just an elite tool. That transition from niche research gadget to mainstream diagnostic, that's where the real public health impact is going to happen.
Absolutely. That's the game changer. Okay, this seems like a good moment to pause for a second. If you're finding this insightful and you're watching on YouTube, please do take a moment to like this video, subscribe to Tomorrow Unveiled, and maybe consider hitting that new hype button. It really helps us reach more viewers. And of course, make sure you turn on notifications so you never miss an update on the future.
And if you're listening to Tomorrow Unveiled on a podcast service, maybe Apple Podcasts or Spotify, we'd be incredibly grateful if you could take just a moment to leave us a rating and a review. It genuinely helps Tomorrow Unveiled reach new listeners who are just as curious about the future of health and longevity as you are.
We really do love hearing your thoughts. So whether you're on YouTube or your favorite podcast app, drop a comment and let us know what you think about the Immortality update so far. What stands out to you? All right, so we've established how we might fuel the cell with things like NMN and how we can measure the results with ever-improving biological age clocks. Now, let's talk about the next level of interventions. This is more like the therapeutic frontier, right? The stuff aimed at actively repairing damage that's already been done. The research our team compiled specifically highlights Senolytics and Telomerase therapies here. The cleanup crew, as you called them earlier.
Exactly. These represent a more aggressive push. It's not just about maintenance or topping things up. It's about actively fixing age-related damage to extend that crucial health span. Let's start with Senolytics. These are compounds, often drugs or drug combinations, designed specifically to target and eliminate senescent cells. You sometimes hear them called zombie cells.
Yeah, zombie cells. It's a catchy term. Why are they so bad? Why zombies? What makes them so damaging that we need specific drugs just to clear them out? Don't they just stop working?
If only it were that simple. It's not just that they stop dividing, which is part of the definition of senescence. The real problem is what they do actively. A senescent cell doesn't just sit there quietly. It doesn't undergo normal cell death, apoptosis. Instead, it enters this kind of metabolic limbo state where it starts spewing out a whole cocktail of highly inflammatory proteins.
Inflammatory proteins.
Yes, collectively known as the Senescence-Associated Secretory phenotype, or FSP for short. Think of the SSP signals like a constant, low-level inflammatory broadcast. It's like a slow-burning fire that damages surrounding healthy tissues, accelerates their aging, and directly contributes to a whole host of chronic age-related diseases. Things like arthritis, lung fibrosis, cardiovascular problems, even neurodegeneration.
So they're actively poisoning the neighborhood, basically.
That's a pretty good analogy, yeah. They create this toxic pro-aging environment, and that's where Senolytics come in. They are chemical agents, combinations often involving drugs like dasatinib, a chemotherapy agent, paired with natural compounds like quercetin, found in onions and apples that selectively trigger apoptosis or program cell death only in these senescent zombie cells.
So they hunt down the zombies and tell them it's finally time to die, leaving the healthy cells alone.
That's the goal, and increasingly the reality in preclinical and early clinical studies. You're literally cleaning out the inflammatory debris, removing these bad actors from the system.
So with Senolytics, you're not just trying to pause aging, you're actively removing accumulated damage. That feels like a really powerful strategy.
It is potentially transformative, yes. If we can periodically clear out these senescent cells, we might significantly delay or mitigate multiple age-related diseases at once.
Okay, that's Senolytics. And the second major therapeutic strategy mentioned was telomerase therapy. What's the story there?
Right. Telomerase therapies focus on the protective caps at the ends of our chromosomes. They're called telomeres.
Ah, yes, the shoelace tips analogy.
Exactly. They protect the coding part of the DNA from degrading when the cell divides. The issue is, every single time most of our cells divide, these telomere caps get slightly shorter.
Like a countdown timer.
Pretty much. When they become critically short after many divisions, the cell gets a signal. It either becomes senescent, turns into one of those zombie cells we just discussed, or it undergoes apoptosis and dies. Telomere shortening is a fundamental clock of cellular aging.
So what does telomerase do?
Telomerase is a specific enzyme. Its job is to add back DNA sequence to the ends of the telomeres, maintaining their length or even potentially lengthening them. Most of our adult cells have very low telomerase activity, which is why telomeres shorten over time.
So if we could somehow boost telomerase activity safely, we could maintain telomere length. We could essentially extend the number of times a cell can safely divide.
That's the hypothesis driving telomerase therapy research. By maintaining telomere length, you could potentially extend the healthy lifespan of cells, maintain genomic stability for longer, and delay the onset of cellular senescence.
Keeping cells functional and dividing healthily for longer.
Exactly. Both Senolytics, the cleanup crew, and telomerase therapies, maintaining the cellular clock, represent these key therapeutic targets confirmed by the clinical reports. They're aimed squarely at pushing the boundaries of health span and, ideally, preventing the onset of multiple age-related chronic illnesses simultaneously, rather than treating them one by one after they appear.
It's a very different approach to medicine. And you also mentioned that this sophisticated molecular profiling we talked about earlier is validating some simpler compounds in this cleanup category too, like spermidine.
Yes, that's another interesting thread in the research. Spermidine is a naturally occurring polyamine molecule. You find it in foods like aged cheese, mushrooms, legumes, whole grains.
Okay, so something dietary.
It can be, yes. And its validation through modern molecular techniques is exciting. The profiling work is confirming that spermidine seems to exert its beneficial effects on cellular aging, primarily by enhancing a process called autophagy.
Autophagy, that's the cell's internal recycling system, right?
Exactly. It's the body's natural way for cells to systematically clean out damaged components. Old proteins, dysfunctional mitochondria, other cellular debris, they break them down and recycle the parts. By boosting autophagy, spermidine essentially helps ensure the cell's internal infrastructure remains high quality and functional. It helps the cell tidy up after itself.
So it's like preventative maintenance for the cell. Helping it clean house regularly delays the buildup of the kind of damage that might eventually require the more aggressive senolytic intervention.
That's a perfect way to think about it. It's an effective strategy for maintaining cellular health and resilience, potentially delaying senescence.
Okay, that gives us a pretty robust picture of the current therapeutic landscape. We're moving from just providing molecular fuel like NAD plus set to measuring progress with biological clocks, and now to these active cleanup and maintenance strategies like senolytics and autophagy enhancers. But now, let's shift gears a bit. Let's talk about the finding in the material that, honestly, I found the most surprising, maybe even profound. It's the emphasis on the importance of early life, like even before birth.
Ah, yes. This is truly a foundational concept, and it's really shifting paradigms. A lot of this insight, as referenced in the sources our team compiled, seems to be emerging from places like the Human Longevity Laboratory at Northwestern University. They're really pushing the timeline back, way back, analyzing how factors related to maternal health can actually program long-term aging outcomes for the offspring.
Right. We always talk about the genes we inherit, our DNA sequence, but this sounds different. It's not about the genes themselves, but about how they're programmed, or how other things are programmed.
Exactly. It's less about the hardware, the DNA sequence, and more about the initial software settings, you could say. The specific factor that the research analyzed is the impact of maternal nutrient restrictions during pregnancy.
Nutrient restrictions, like the mother not getting enough food?
It could be famine or severe malnutrition, yes,
Right.
but it can also encompass periods of significant metabolic stress, perhaps severe illness, or major nutritional deficits, even in generally well-nourished populations. What the findings suggest is that these conditions during pregnancy can profoundly influence the offspring's aging trajectory down the line, and it happens via something called mitochondrial programming.
Mitochondrial programming. Okay, mitochondria are the powerhouses of the cell, right? They generate energy.
Correct. And critically, mitochondria contain their own small amount of DNA, and they are passed down almost exclusively from the mother through the egg cell.
Okay. So the mother's health directly impacts the mitochondria the child inherits.
It seems to go deeper than just inheritance. The environment during gestation appears to program how those inherited mitochondria function long-term. If the maternal environment signals stress or scarcity, not enough nutrients available, the offspring's mitochondria seem to adapt in utero.
Adapt how?
They might become sort of hyper-efficient in conserving energy, programmed for a world of scarcity. But this adaptation can come at a cost. It might lead to them being structurally compromised or more prone to dysfunction later in life when, perhaps, nutrients are abundant. It creates this kind of legacy effect.
A legacy of potentially inefficient or dysfunctional energy processing that lasts throughout the child's entire life, programmed before they were even born.
Precisely. Even if that child later grows up eating a perfect diet and living a healthy lifestyle, their fundamental cellular energy machinery might be compromised from day one because of that early programming.
That is profound. It really reframes where health and aging begin. What are the actual long-term health implications? Does the data confirm specific risks linked to this mitochondrial programming?
Yes, the research confirmed a dramatic and measurable increase in the offspring's risk much later in life for serious, typically age-related conditions. Specifically highlighted were cardiometabolic diseases, things like type 2 diabetes, metabolic syndrome, heart failure, and also chronic kidney diseases.
Wow. So things we usually associate with lifestyle choices in middle or old age could have roots stretching all the way back to the prenatal environment.
That's what the evidence strongly suggests. It really proves that long-term health and even the fundamental pace of biological aging can start being set well before birth. And it underscores something quite powerful. Optimizing the maternal environment, ensuring good nutrition, reducing stress during pregnancy might actually be one of the most potent non-pharmaceutical longevity interventions we currently know of. It's about setting up the next generation for a healthier, longer life from the very beginning.
Okay, these breakthroughs we've discussed, the NAD plus boosters, the Senolytic cleanup crews, the personalized biological age testing, and now these profound insights into early life programming, it's all genuinely thrilling. It offers so much potential. But it brings us back to that practical question. If the data on things like NMN, Senolytics, and personalized testing is becoming so robust, why aren't these approaches already standard practice in hospitals and clinics worldwide? What are the big institutional hurdles? What's slowing down the transition from lab bench to bedside?
Yeah, that's the multi-billion-dollar question, isn't it? And it's the essential structural challenge that was identified across the clinical reports and industry analyses our team looked at. The core issue stems from the very nature of longevity science itself.
How so?
We are, by definition, studying biological processes aging that unfold over decades. Yet our entire research funding infrastructure, our logistical models for clinical trials, they are overwhelmingly built for short-term projects.
Right. I can only imagine trying to secure funding for a study that needs to track thousands of people for, say, 25 or 30 years. It must be nearly impossible.
It is incredibly difficult. Funding limitations are consistently cited year after year as the #1 barrier. Current institutional and governmental funding models heavily prioritize projects that can yield publishable data, maybe a patent, within 2 to 5 years. That model just doesn't work well for longevity.
Because you need decades to prove something really works long term.
Exactly. the sustained multi-decade funding required to track the true long-term impact of a molecular intervention to definitively prove, for instance, that taking NMN from age 50 actually delays the onset of heart disease or cognitive decline by 10 or 20 years compared to a control group. That kind of funding is incredibly hard to secure and maintain. It creates this huge research gap between promising early findings, the proof of concept, and the large-scale long-term clinical validation needed for regulatory approval and widespread adoption.
Okay, funding is a massive hurdle. And even if, somehow, you did manage to secure the funding for a 30-year study, keeping the participants involved and compliant for that entire time, that sounds like a logistical nightmare, too.
You've hit on the second major issue, participant retention. It's unavoidable in long-term studies. People move houses, they change countries, they change their lifestyles drastically, they develop other health conditions, they simply lose interest, or, sadly, they pass away from unrelated causes.
The dropouts are inevitable.
Highly inevitable. And high dropout rates over these long periods are a serious problem. When subjects leave a study prematurely, it leads to missing data, inconsistent follow-up, and it really undermines the statistical power you need to validate a finding conclusively. You end up with results that are suggestive, maybe, but not definitive enough for broad clinical guidelines.
And are there ethical considerations slowing things down, too?
Yes, absolutely. The research team noted ongoing ethical considerations as another significant factor. There are, quite rightly, strict regulations and ethical reviews that restrict how rapidly and how widely certain novel interventions can be tested, especially in diverse human populations. This is particularly true for interventions that involve more advanced approaches like genetic manipulation or complex cellular therapies. Ensuring safety and equity takes time and careful planning.
Okay, so we have funding cycles that are too short, logistical nightmares with participant retention, and necessary ethical safeguards. The research community seems pretty unified on identifying these challenges. Is there a similar consensus emerging on how to actually fix this, how to standardize the field and accelerate the validation process?
Thankfully, yes. There is a powerful and growing movement pushing for standardization. This is apparently a key discussion point at high-profile industry gatherings, like the Longevity Med Summit mentioned in the materials. The primary goal everyone seems to be rallying around is the need to standardize data collection protocols.
Standardizing data collection, what does that actually mean in practice?
It means getting everyone in the field, researchers, clinics, even diagnostic companies, to agree on using the same language, the same tools, the same metrics when they conduct studies or measure outcomes.
So she's using the exact same test for biological age, for example.
Precisely. It involves implementing uniform measurement protocols. For instance, agreeing to use the exact same, perhaps proprietary, assay for DNA methylation profiling across multiple international trials, or agreeing on a specific defined panel of blood markers and calculation methods for determining metabolic health status.
Why is that so critical?
Because if Clinic A in California uses one epigenetic clock, and Clinic D in Germany uses a completely different one, and Clinic C in Japan uses a third, we can't reliably compare their results. We can't pool the data effectively. We can't build the truly vast, diverse, long-term data sets that we need to definitively prove what works for whom and under what circumstances. That kind of large-scale data is essential for convincing regulators and changing standard medical practice.
So standardized protocols are like building the essential infrastructure, the operating system for global longevity medicine research.
That's a fantastic analogy. Yes, it's about creating the common framework needed to enable large-scale collaboration and accelerate scientific progress through shared comparable data.
Okay, so looking ahead then, based on all the 2025 data points our team gathered, what is the final roadmap? What are the key future goals for the longevity field?
The roadmap, as corroborated across the sources, seems pretty clear on the main priorities. First, there's a continued relentless push for even better biomarkers of aging. We need tests that are more precise, more reproducible, more sensitive to change, and crucially, more affordable and accessible than the current generation.
Better measurement tools first. Makes sense.
Second, there's a strong commitment to conducting future studies that involve increasingly diverse populations. We need to ensure these interventions are safe and effective for everyone across different ethnicities, genders, socioeconomic backgrounds, not just effective for the relatively narrow demographic groups that have often dominated early trials. Equity is key.
Absolutely critical. And third,
it's really about establishing and implementing those standardized clinical protocols we just talked about. The goal is to make personalized aging interventions, assessment, monitoring, treatment, something that is accessible, reproducible, and verifiable, outside of just a few elite research labs or high-end clinics. It's about bringing this science reliable to the community.
So better biomarkers, more diversity in studies, and standardized protocols for accessibility and collaboration. It sounds like the field is really maturing, moving towards rigor and reliability.
That's definitely the trajectory.
This whole deep dive for Tomorrow Unveiled, the Immortality update, it really shows that longevity isn't just science fiction anymore. It's rapidly becoming a field of sophisticated, measurable, and increasingly actionable science. We seem to be moving decisively from just hope and hype towards objective, personalized data.
It's an exciting time, for sure. The transition is palpable. #outro. So, if we were to synthesize the core takeaways from this Immortality update, I think it's clear the longevity science in 2025 has really embraced clinical rigor. We see this movement driven primarily by, well, two main pillars of intervention. First, enhancing cellular fuel and energy, particularly through NAD, plus boosting strategies involving compounds like NMN and Resveratrol.
Right, fueling the engine.
Exactly.
Yeah.
And second, developing targeted cellular maintenance and cleanup strategies, using therapies like Senolytics to remove damaged zombie cells and compounds like Spermidine to enhance natural autophagy or cellular recycling.
Repair and maintenance.
Precisely. And underpinning all of this progress is the parallel advancement in measurement tools. Increasingly precise ways to track biological age, particularly through epigenetic profiling, and a deeper understanding of systemic factors like metabolic health from blood tests, and the crucial role of the gut microbiome. It's all steering us firmly towards truly personalized, data-driven medicine.
And as we move forward, that whole concept of personalized measurement, especially thinking about biological age, it brings us to a really challenging final thought for our listeners. This builds directly on that discovery about early life influence that we discussed.
The mitochondrial programming piece.
Exactly. We learned that long-term health risks, the very pace of biological aging, can be significantly influenced, even programmed, by factors like maternal nutrition and stress impacting mitochondrial function, literally in utero.
The legacy effect of our origins.
Yeah.
It's quite profound.
So, consider this scenario. What if, in the fairly near future, we become able to accurately measure and predict an individual's lifetime disease risk decades, maybe even generations, in advance? Imagine, say, based on a detailed analysis of a child's epigenetics and their mitochondrial profile taken shortly after birth.
A predictive health horoscope, almost.
Kind of. What moral and ethical responsibilities does society assume with that kind of knowledge? If that data tells us, with high probability, that a particular child has a significantly elevated risk for developing severe cardiometabolic disease by age 40, purely because of their mitochondrial programming inherited from a period of maternal stress, where should the intervention begin?
That's a heavy question.
Right. Is the responsibility on the individual child, maybe mandating certain early life treatments or lifestyle protocols for them? Does it fall on future parents, perhaps involving aggressive genetic or epigenetic counseling before conception? Or does it demand a complete restructuring of public health policy and funding, making things like universal prenatal nutrition support and stress reduction programs for expectant mothers the absolute top priority?
Where does the societal obligation lie when we can see the risks so far ahead?
Exactly. The ability to predict health outcomes with such granularity so far in advance, based on factors set before someone even makes their own choices. It creates this immense ethical mandate, doesn't it? A need for proactive, systemic public health policies that we're only just beginning to grapple with.
The weight of that foreknowledge.
Yeah.
It's a fascinating and complex challenge, and it will undoubtedly define the next phase of personalized longevity in public health.
Indeed it will. Something profound to think about. Thank you for joining us for the Immortality Update. We promise more in-depth exploration of where science and society are heading next time on Tomorrow Unveiled.